So, there will be questions about it. Anyway, I just wanted to review a little bit. You know, this xylem stuff was a lot of review already. But then the last topic that I talked about was this paper in Science where they looked at the hydraulic conductivity of sections of xylem and found that it was not constant, even though the conducting cells in the xylem are dead when they're mature and they're functional, they're dead. But even though they're dead, the conductivity isn't a constant. And they did this by taking little sections of stems and putting tubes on them that they could put a pressure differential across, right? So delta P, P1 minus P2, doesn't matter. Okay, so you put a pressure differential across this and measure a flow rate of stuff through there. And this flow rate is equal to the difference in the pressure is divided by some resistance. There's some hydraulic resistance or the inverse of the hydraulic conductivity. And by measuring the flow rate and knowing what pressure differential they put across the stem, they can solve this equation for what the resistance is, right? If I do this right this time, right, something like that. So they can calculate what the resistance is of a little piece of stem or a little section of xylem. And they did it, and what they found out, just to reiterate, oh, there we go. Just to reiterate what happened. When they used distilled water, they got a lower conductivity, a lower hydraulic conductivity, a higher resistance with distilled water than when they used water with some nutrient dissolved, some ion dissolved in that. And potassium chloride is here. So when they used concentrations of potassium chloride that are found typically in the xylem, they got a smaller resistance than when they used pure distilled water. And it puzzled them because these cells are dead. How can this be? And they took a close look. They also did it with an intact plant, a living system, not just pieces of stem, but they did it with living system, found the same results. And they looked at a bunch of different plants and found similar results across, what did they say? 19 different angiosperms, 5 conifers, and 3 ferns. So really different plants all showed this response. And to cut to the chase, what they decided was that the pits between vessels, so you have a xylem vessel, inside the xylem vessel like a hollow tube, but the vessels are connected by pits. And the pits are also openings, no membrane but holes, but they're just very, very small holes that connect the vessels. So when they were able to use a piece of a stem that only had one xylem vessel, they didn't see this dependence on the concentration of ions. It depends on flow rate. But if the vessel diverged, or if there were two that were connected by pits, then they noticed this funny behavior. And so their conclusion was that these pits shrink and swell. The diameter shrinks and swells depending on the composition of the fluid that's flowing through it. And with distilled water, the pectins that are found in the cell wall swell up and make this opening smaller. And so the resistance is larger. And with a little bit of an ionic solute, these pectins shrink, make the pit bigger, the opening bigger, and the resistance smaller. So even though these cells are dead when they're functional, their properties aren't necessarily constant. There's still some dynamics going on. So this is one example. The other example that I talked about was, if you get an embolism in the xylem, that really affects the hydraulic resistance because it totally blocks off that pathway and the water has to find another way around to get through. And so embolisms also cause changes in hydraulic resistance of the xylem. But that's a dramatic thing. This is a more subtle change. Okay, so that's where I wound up, and then I said there were more slides after the supposed ending. So let's talk about this for a little. Let's talk about wine. Here are two kinds of wine grapes. This one's called Grenache. This one's called Chardonnay, right? Maybe you've heard of them. They're both Vitis vinifera, right? The most common type of grape for making wine. But they have different behavior, especially under drought stress. So this variety, Grenache, is more drought tolerant than this one, Chardonnay. Not surprising, there are differences amongst genotypes for drought tolerance for many plants. Grenache is said to be more pessimistic under water stress. What it means is when this plant gets water stress, it closes its stomata, so it doesn't lose much water, right? When this one gets stressed, doing the anthropomorphic thing, when this one gets stressed, it decides, I better hold on to all my water because I may not get any more water. So that's why it says pessimistic, right? Versus Chardonnay, which is optimistic, right? It says, I'm running out of water. It'll rain tomorrow or whatever, so I don't have to worry about saving anything. So it's optimistic with the water stress, right? Okay. This one has a lower average transpiration rate per unit leaf area, this one higher. And so this combination of traits is called, for Grenache, the pessimistic one, the one that closes its stomata right away, is called isohydric. Whereas the Chardonnay is called anisohydric. So isohydric, the same water, it tries to keep its amount of water the same. This one, anisohydric, water level fluctuates all over the place. All right, so just two different strategies within the same species from these two different cultivars. And what this group did was they were interested in that behavior and what was different about these two plants that made them behave this way, particularly under water stress. So they looked at the hydraulic conductivity of the root system. So L sub 0 is an indication of the root hydraulic conductance divided by the root dry weight, just to normalize it and compare different samples. And here is that root hydraulic conductance normalized to root dry weight versus time of day for the Chardonnay plants. And you can see that start out somewhat some low value and increases at midday and then decreases. And a statistically significant increase in the root hydraulic conductance in this midday period. Okay. This root hydraulic conductance is also, in large part, the magnitude of the root hydraulic inductance is related to the transpiration rate. So it's the root properties in many cases that are controlling the amount of water going through the plant, or at least as much control as the stomatal properties. And here's this relationship for both cultivars on the same graph, and noisy data, but it's not too bad. Okay, so but the interesting thing is that this root hydraulic conductance is not constant over the course of the day. All right, so looking at this. Okay. They looked at this change in the hydraulic conductance between early in the day and at midday. So A and B, those 2 points. And that's shown on this graph. So here's early in the day, later in the day. And you can see for the Chardonnay control, this one, sure enough, it has an increase in this hydraulic conductance at midday compared to the morning. Same thing happened in Grenache, increase later in the day for the control plants that were not water stressed. For the water stress plants, Chardonnay, okay, so the water stress reduced the hydraulic conductance of the root system both in the morning and the afternoon, but the relative change was about the same for Chardonnay. Whereas for the Grenache, the one that's pessimistic, that tries to save its water, its root hydraulic conductance did not increase at midday when it was water stressed. So the statistics, you can see effects of time of measurement and water stress were significant for both cultivars, but there was the interaction term was only significant for this Grenache one. So it had different behavior on the water stress than on the non-water stress. All right? If you want a step further than that, I mean, this is interesting already. Good stuff. Okay. They measured the hydraulic conductivity of single cells using I assume, I don't remember, a pressure probe like we talked about. And so they poked that into cells in the root cortex outside the endodermis, in the cortical, or in the root cortex. And they determined the hydraulic conductivity of single cells. And what is the control and water stress? Okay, control water stress for Chardonnay, control water stress for Grenache. This is the pessimist that hangs onto its water. No change, no statistically significant change in root conductivity, in single cell conductivity with water stress. But with Chardonnay, when they were water stressed, these cells increased their conductivity, hydraulic conductivity. That's interesting. So then that raises the question, how did they increase how did this happen? How is the plant able to do it? So they looked at cells in the root, and we don't have to spend a lot of time on this, but they looked at where suberin was deposited within the areas between the cells, both in the ectodermis and the endodermis, and they saw whatever they saw. Well, whatever they saw was there's a lot of suberin around, and water has a hard time going through there, particularly in the endodermis. All right. Then they looked at the expression of these two genes. These two genes, VVPIP11 and 22, are both aquaporins, and they're found in the plasmalemma of these cells. VV, Vitis vinifera, that's the genus. PIP, plasma intrinsic protein. That's what they called aquaporins before they realized what their function was. So VVPIP11 is one aquaporin. Here's a different aquaporin. They looked at gene expression. Here for Chardonnay, well watered, they see changes in the 1,1 gene, in gene expression. In fact, a doubling of the relative expression relative to some other thing in the genome. Right? A doubling. The other aquaporin didn't change, so that's not the story. But this one, actually double at midday compared to early in the day. Here you can see it on a bar graph. Control level of expression, drought stress, significant increase in the expression of this aquaporin gene for Chardonnay, the one that's the water waster. So it's doing some work to waste that water, right? It's expressing this gene more and more and more to maintain transpiration under drought stress. The two things didn't change, didn't matter. And so their conclusion was that aquaporins have a lot to do with the hydraulic conductivity of the root, not so much the longitudinal conductivity, once you're in the xylem, because there's no membranes or anything. So that's not, we're not looking at that, but this other experiment I just talked about. But more the radial conductivity from the soil into the xylem. And by increasing that radial conductivity, they could affect the hydraulic conductivity of the whole root system. And that's what allows this variety to maintain transpiration rates under water stress, where this one reduces its transpiration rate under stress conditions. So aquaporins have a big role to play in hydraulic conductivities. It happens at the cell level, the plasma membrane, where they're measuring these things, but it affects the conductivity of the whole root system. And it leads to these different behaviors. Isohydric, trying to hold on to the water, same amount of water, or anisohydric, where things move around.
So which one of them had the Subarus?
They both did.
Oh, both
Yeah,
did.
they both. And you can look at this, at your leisure, you can look and see, well, this one have more than the other, I don't know. But yeah, they both have suberized areas. So anyway, just tying together some of this root behavior in terms of hydraulic conductivity with our earlier talks about membranes and aquaporins and how plants can regulate those hydraulic properties. That's what's at the end of that set. Sometimes I don't talk about this, but I thought since we're moving right along this semester, I have time to talk about some of these things. Sure.
So the radial conductivity was in the isohydric plant, right?
I mean, it's in both. It's in both. Okay, so the ease with which water can move from the soil into the xylem and up the stem of a plant. The resistance of that pathway is determined by the resistance of the xylem itself and the resistance of the root from getting out in the soil into the xylem. And since we have the endodermis with this highly suberized region, the water has to cross the membrane to get into the xylem from the soil. And so that's the point where the plant can change the expression of these genes and affect how easily, it can affect that radial hydraulic conductivity. And it's a combination of the radial and this axial conductivity that determines the whole.
So the isohydric has better control, is that what, or it expresses that one gene?
Control of
over the
what?
conductance? Radial conductance?
This is the one actually that changed its radial conductance. This one said, oh, it's stress. I better express more of that gene and allow more water in here so I can maintain my transpiration rate. This one says, I'm just going to reduce my transpiration rate. I don't care about that. I want to save water. This one says, my strategy can work too, where I don't.
So it changed the radial conductivity to let more water.
Right, that's what this one does, and that's how it can maintain its transpiration rate even in the face of water stress. Right, so it's less drought tolerant, because if that stress is prolonged, this one's not gonna make it. This one maybe will. So, yeah, it's kind of counterintuitive. Why would the plant go to all this extra work to do something that's not gonna really maybe pan out in the long run? You know? But in the short term, this one's functioning and photosynthesizing probably more than this one is, because this closes the stomata and then it can't get CO2.
So, can I say that the water intake is bigger in this one's in drought stress, like in the Chardonnay one?
Yeah, under drought stress, I would say the water intake would be bigger for this one. Yeah,
Okay.
I think so. Yeah, higher, and so this results in higher transpiration rates per unit leaf area for this one than this one, because this one, when it gets stressed, closes stomata, lower transpiration. Yeah. That's kind of interesting. So, and these terms are applied not just to grapes, but to any plants, isohydric and isohydric. Those are general terms that are used. Sunflowers tend to be anisohydric. They just pump out water until there's none left. Whereas something like maybe soybean was more isohydric, closest stomata. No, I don't know. But sunflowers act this way. A lot of other plants. So there you go. Again, I guess emphasizing the point that these plant hydraulic properties can change. Even after the vascular system is formed, even after it's in place, those hydraulic properties are under some control by what the plant is doing. All right. Moving right along. Let's talk about flow. I talked about xylem, now we talk about flow. And so, first, start with sort of a review of what's in the phloem, the cells that make up the phloem, how they're arranged, and what they look like, and things like that. And then, after that, I'll talk a bit about transport in the phloem, and loading of solutes into the phloem, and unloading of solutes out of the phloem. That's where we're going. But for now, let's start with a general review of what is happening in the phloem. Okay. So, unlike the xylem, the conducting cells of the phloem are alive when they're mature and they're functional. Dead cells and living cells. The different types of cells making up the phloem tissue, sieve cells or sieve tube members. These are the cells that conduct the stuff in the phloem. Companion cells, which are affiliated with these cell types. And then fibers and parenchyma, just like in the xylem. Fibers for structural support, parenchyma, and unclear. Okay. Sieve cells. Sieve cells are the conducting cells in the phloem that are found in gymnosperms and ferns, pteridophytes. Sieve tube members are found in angiosperms. Analogous cells in the angiosperms that conduct things are called sieve tube members. And why sieve? Because you know what a sieve is, like a screen, like a soil sieve for separating particles. And sieve cells tend to have areas that look like screens in them. You'll see some Okay. Collectively, these conducting cells are called sieve elements, and their name comes from parts of them look like a sieve. Sieve cells are connected via sieve areas. So you have one sieve cell, another sieve cell, and the connection between the two is called a sieve area. Sieve tube members also are arranged end to end, and the connections between sieve tube members are called sieve plates. In both cases, these connections are made up of holes between the adjacent cells. Holes with no membrane over them. Open holes. And it's just that one difference between these is the holes between the sieve cells tend to be a little smaller and not as elaborate, I guess, as the holes between the sieve tube members, which are bigger and offer less resistance to movement of fluid through them. Okay, sieve elements, the length from a tenth to several millimeters long. And the lumen radii, the open area in the middle of the cell, 6 to 25 micron radius. So similar to what's in the xylem. Sieve elements are alive. So these cells, these conducting cells, are living cells, but they don't have a nucleus. How can they be alive without a nucleus? Good question. They have cytoplasm, and they have a plasmalemma, so there's a membrane around these cells. The membrane is continuous from one sieve element to the next. In other words, it doesn't cross that hole, but it goes on the outside of the hole from one cell to the next. And I'll show you some pictures. Sieve elements don't have tonoplasts, they don't have vacuoles. They don't have nucleus, they don't have mitochondria. How can they be alive? Good question. Okay. So, sieve tube members are connected end to end to form sieve tubes, and these connections are called sieve plates. They're about, so here's a sieve tube member, here's an adjacent sieve tube member, here's a sieve plate, here's another sieve plate. These sieve plates are about 5 microns thick, and they've got a lot of holes in them. So if you look, you can, you see this angled end of the cell with a bunch of holes, wide open holes. So the plasma membrane of this cell is along the outside of the cell. But up here, it goes on the periphery of that hole to the next cell. It doesn't cross the hole, it doesn't block off the hole. There's no membrane across any of these openings between adjacent sieve tube members. The holes, the pores, the holes are about, 1 to 3 microns in diameter. And in many or most cases, these sieve plates are angled relative to the axis of the cell, so that if you add up all these areas here, there may be more area than the cross-sectional area of the sieve tube member itself, because it's on an angle, it's got a lot of holes in it. So it offers a pretty low resistance pathway for the movement of water. I mean, not entirely, it's not a superconductor, it's got some resistance, but it's not as big of a resistance as if this was a flat across. Okay, so here's a closer picture of a sieve tube member here with the sieve plate. You can sort of convince yourself there's holes in there. And the membrane system from one cell is continuous to the next. All right. Those are sieve tube members. And sieve, okay, the other thing is this lateral sieve area. So sieve areas, if this wasn't an angiosperm, if this was a gymnosperm, these connections would be called sieve areas, and in general they would have smaller holes, but still be openings between adjacent cells. Just smaller and not as elaborate, and convince yourself that the gymnosperms and the pteridophytes are lower vascular plants, whereas the angiosperms, those are the high ones. They've really evolved efficient mechanisms, right? Okay. Okay. So, but even in the angiosperms with sieve tubes and sieve tube members, adjacent sieve tubes can also have connections between them called sieve areas, lateral sieve areas, in the case of the angiosperm. So there is some communication between adjacent sieve tubes. All right. A lot of times, here's a close-up of a sieve plate with here's the cell wall part, and here are the pores, and this stuff labeled with a P is found here. These are proteins that are found in the pores of the sieve plate itself, and it's called P protein, from phloem protein, stands for. Many plants have this, not all plants do, but many plants have this P protein, particularly all dicot species, a lot of monocot species. So here's the plant that this image was taken from. And its function is not quite clear, but it probably has something to do with repairing damage to the sieve tube. So if something comes along and cuts into the sieve tube, it's in the plant's best interests to plug up those holes quickly and not lose a lot of stuff out of the phloem, because the phloem is a relatively concentrated solution of, in a lot of cases, sucrose. So the products of photosynthesis, what the plant's working on to fix carbon and to be productive. So it's not in the interest of the plant, if something damages us, to have that leak out. And if the damage does occur, it's thought that these P proteins may help in blocking these open holes quickly, very quickly. Here you go. Last sentence. This is kind of giving it away. P proteins are synthesized in the companion cells and transported to the sieve elements via plasmodesmata, which starts to answer my question: how can the sieve element be alive if it has no nucleus, no mitochondria, no vacuole? It's got a companion cell next to it, and the companion cell is loaded with mitochondria. It has a nucleus. It's alive. These cells are called albuminous cells for the gymnosperms, but that's the same idea. What's more,